In this lecture, we're going to explore a very briefly explanation about flow cytometry and automated cell contrast, specifically in respects to this instrument we have in lab called Moxi V, and we'll be using it in our lab to count cells. So what is flow cytometry? Flow cytometry is a technology that provides rapid analysis of single cells in a solution. Flow cytometers use lasers as light sources to produce both scattered and fluorescent light signals that are read by detectors such as photodiodes or photomultiplier tubes. Cell populations can then be analyzed or purified based on their fluorescence or light scattering characteristics. So basically, we can detect different cell populations within a cell culture, and then we can analyze those. We can purify those different populations, and we usually can do these by using fluorescence or light scattering characteristics. So we'll be focusing with Moxi V on light scattering characteristics. Cells have what we call clusters of differentiation, or CDs. And these represent different receptors or different proteins on the cell surface of a cell membrane that allow biologists to distinguish between types of cells. Over 400 different CD markers have been identified. So here on the right, what you can see are clusters of differentiation, which are proteins on the cell membrane that define the type of cell we're working for. So each cell, each different cell type, will express different proteins on the membrane. Some of these proteins are either receptors or ligands. So we can think of the keyhole and key model here. If you are a receptor, things will bind to the keyhole, and the receptor will trigger this cell, this specific cell, to do something. If you're a ligand, the key, you can bind to a receptor on another cell and trigger that other cell to do something. We can use these CDs to differentiate cells using flow cytometry. So before we talk about flow cytometry, we're going to have a brief review about light, which is the basis for flow cytometry instruments. So we can all see colors based on the wavelengths given out by a specific product. So the product's what gives out the wavelength, and so we see different things in life with different colors. So for example, when we see a plant, light is hitting the plants, but the plant only reflects back the wavelength between the 500 nanometer wavelength. And so we see plants as green. So what wavelengths really refer to is the distance from one peak of a wave to another peak of a wave. And the light reflected on those waves is going to be of different colors. So with flow cytometry, what we do is we make fluorescent markers. These are molecules that will light up when hit by a laser or excited on a specific wavelength. The color they change off is called the emission color. So scientists have used the knowledge about light to make these fluorescent markers. Compounds, again, then when these are hit with a specific wavelength, they will emit a specific color that we call an emission color. We can detect these, and we call these fluorophores. So we can use these along with what we know about cells, and we can use these to detect specific characteristics about cells. So here you can see a list of different fluorophores. The color, the fluorescent color they emit, the range of the wavelength that we can use to excite them, and the relative brightness. So how useful these are in a lab. Scientists can use these fluorophores to mark CD markers on cells, so to mark those peripheral proteins, these proteins of the cell membrane, and then be able to recognize the specific cell types. So for example, here you have a CD25 and a CD3. So these are two different markers, okay? These markers can be tagged or can be found with specific antibodies, okay? So we can use antibodies that can bind these markers, and then the antibody itself, as you can see in these two figures, will be bound to a fluorophore. Remember that antibodies are molecules that have specific affinity to certain CDs. There are specific antibodies, for example, to both CD25 here and CD3 here. So these antibodies is specific for CD3. This antibody will only bind CD25. Therefore, in this example, after doing flow cytometry, we can detect both red and green lights, and then we can then say, okay, this cell, if it's emitting both green signals and red signals, must be an activated T cell, since only T cells have these CD25 and 3 markers, and hence it must be a T cell. Remember, a T cell is a cell that is part of our immune system. An application of flow cytometry is culture counting. Culture counting is the reference method throughout the world for particle size analysis. So we can use culture counting to analyze the size of particles. The culture method of sizing and counting particles is based on measuring changes in electrical impedance produced by cells and cellular particles. So cells in solution pass through what we call a sensing zone. The displaced volume is then measured as a voltage pulse. So the cell is going to impede this voltage, this displaced volume. And the pulse is proportional to the volume of the particle. So the bigger the pulse, the bigger the cell. Moxi V is a type of cell culture counting machine. So here's a schematic. You have 2 electrodes that are going to create a gradient here. The cells, one by one, will pass through the gradient and disrupt this electric gradient. They will block it. The bigger the cell is, the less voltage that will be able to pass through the gradient, and so the bigger the block. Therefore, the bigger the cell, the bigger the disruption. So the pulse received by the machine is proportional to the volume or size of the cell. Using this method, we can detect cell size and also population in a cell culture vial and viability of these cells. Moxi V specifically works with this type of cassette that you'll be using in lab. This allows for accessible flow cytometry as it makes flow cytometry affordable and so accessible. It allows us to detect subpopulations, so it can detect 10 nanometer sized resolution, and that's going to allow you to easily identify subpopulations of cells. So how pure is your cell sample? It also allows for simultaneous sizing, phenotyping, and viability counts. A 40 micrometer sensing zone enables users to simultaneously analyze cells for size and viability. So how much of your cell population is alive? And it is also a high volume cell analyzer. The flow cassette enables analysis of 2,500 cells per second, therefore eliminating the use or the need for triplicate counts, like you have been doing with your hemocytometer. The basic principle of the cell counting was combined with flow cytometry using breakthrough technology in this disposable cassette, shown here on the right. This cassette is the basis for the Moxi family of instruments, and it allows users to count 10,000 cells per second. The sizing accuracy of counters enables users to identify cells that are relatively close in size. So how does it work? The way the cassette works is shown here. So we excite the fluorophore with a laser. So here they're showing you the blue wavelength. The laser will pass through the channel on the cassette. So this is on the cassette right here. So here's the light, comes here. Here is passing through the cassette where the cells will also travel through. So here's also where the cells will travel through. The cells can then emit color if they have been treating with fluorophores or propylium iodide, which we'll talk about in a second. And now note that you also have here an electric gradient here, and you will be able to decipher the phenotype of the cell, the size, and the viability. So what is propylium iodine? Propylium iodine is a chemical that becomes very bright red in color when it binds to cytosolic DNA. Propylium iodine is a compound we use to detect dying cells. So shown here, propylium iodine. If you have any cell and it starts to die, the cell membrane starts to become permeable. So here you have an unhealthy cell and you can see the holes on the cell. We can incubate cells with propylium iodine for about 2 minutes, and the dying cells will absorb the propylium iodine. When we sort our cells, we can identify cells that are viable by looking at how red the dead cells emit the red color versus the non-red cells, the healthy cells shown over here, as they will not absorb the propylium iodine. The output of Moxi V is a graph where the cell size is on the x-axis and the y-axis we're going to be looking at brightness, so the propylium iodine. If the cells are bright or red, we can then identify the dead cells from the healthy cells. Here is a quick overview of the actual machine. So what you can see here is that the Moxi V has a USB cable port, so we can output our data and analyze it at our own convenience. Here is the power button. It has a touchscreen display, so it's quite easy and user-friendly. And then here is the cassette tray door, where we will be inputting our samples in our cassettes, and then the samples, the cassettes go into the Moxi V. Okay, so the machine runs quite fast, so this is the output that you'll get once your samples run. So you can see your data in one or two ways. So here on the left, you can see results from the cell counter, and you can see that we can identify the total number of cells, the percent viability, and the diameter of our cells. You notice that the diameter of the dead cells, shown here in red, will be less than the diameter of the living cells, shown here in green. And then in yellow, you have the overlap of both the dead cells and the living cells. It makes sense that the dead cells have a smaller diameter, as when the cell dies, they become permeable, and so they start to shrivel as the water leaves the cell. On the right of this figure, you can see different types of analysis where you can graph the propylium iodine against the diameter of the cell. Here you have the propylium iodine on the x-axis, on the y-axis, and here you have the diameter on the x-axis. Note that the higher the concentration of the propylium iodine will indicate or show you the population here in the scatter plot, the population of the dead cells, as they have absorbed the propylium iodine. Also note, once again, that the living cells shown in this population down here will have a bigger diameter than the population of dead cells. We will be using the Moxi V later in lab.